Okay, first and foremost, so I don't think it comes as a surprise to you, but there are three pillars of biodiversity. So there is genetic diversity, species diversity, and ecosystem diversity. And genetic diversity, of course, is important to both maintain and restore evolutionary potential, fitness, and ecosystem resilience. But without monitoring, sorry, and indicators, we can't really track the processes that threatens genetic diversity, which could be loss of genetically distinct populations, reduced population size, human-induced selection, for instance, due to climate change effects, spread of alien populations, etc. Indicators I mentioned is, and when we talk about indicators for biodiversity, they are used to measure and monitor the state of biodiversity, to identify both pressures and threats, and also to assess the effects of protective measures and restoration efforts. So I will take you back in time a bit. To talk about genetics in both conservation policy and research. So I won't go into all of the details here, but I will dive into the Convention of Biological Diversity, the so-called CBD, in a second. But I would also like, from this slide, to pull out both some of the more recent initiatives, such as the ICN SSC Conservation Genetics Specialist Group, but also a past EU project called Congress Genetics, and also IPBES, which is the intergovernmental science policy platform on biodiversity and ecosystem service. So let's dive in to the CBD a bit. So the first text for the original kind of convention text from 1995 of the CBD has three articles which are worth noticing. Articles 7, 8, and 10 saying identification and monitoring, in situ conservation, sustainable use of the components of biological diversity. So all of the parties of the CBD should both identify components of biological diversity. They should monitor through sampling and other techniques the components of biological diversities, and they should identify processes and categories of activities which are likely to have a significant adverse impact on the conservation and sustainable use of biological diversity and monitor their effects. So over the years, there have been a lot of additional progress in terms of getting genetics and genetic diversity into the CBD global biodiversity frameworks. What I would like to highlight is the past strategic plan for biodiversity for the decade 2011 to 2020. And the Aichi targets, and explicitly target 13, which was the first time that genetic diversity really got into the global biodiversity framework. But the text said that genetic diversity should be monitored and both safeguarded and also improved for cultivated plants, farmed and domesticated animals, and of their wild relatives. But nowhere in the text it said something about wild species. So looking into the more recent initiatives that has been, it's worth highlighting both GeoBon, but also the EU Cost Action Network GBIKE, so Genomic Biodiversity Knowledge for Resilient Ecosystems, and the Network of Networks, the Coalition for Conservation Genetics. Those, along with a lot of other initiatives and also research, has helped push for the awareness of genetic diversity in conservation. So as I mentioned before, the CBD text for the past decade did not include genetic diversity of wild species. And I would like to pull out the paper by Linda Leiker and colleagues from 2020 in Science, which put a focus on the fact that the CBD, Global Biodiversity Framework, did not include goals for genetic diversity of wild species. So that led to a lot of great people putting their minds and efforts together and trying to push for genetic diversity of all species being recognized under the CBD. And the first drafts for the Global Biodiversity Framework that we now have, in effect, did not include genetic diversity of all species. So there was a lot of both research papers, policy briefs, but also a lot of lobbying and awareness going on from these people in terms of raising awareness of the need to include wild species in the Global Biodiversity Framework of the CBD, but also having indicators on genetic diversity. And so three indicators, pragmatic indicators, were proposed to go into the Global Biodiversity Framework of the CBD. So there was indicator 1, which was the proportion of populations within species with an effective population size greater than 500. So effectively looking at genetic diversity within populations. Indicator 2, the proportion of populations maintained within species. So effectively looking at genetic diversity between populations. And indicator 3, the number of species and populations in which genetic diversity is being monitored using DNA-based methods. So fortunately, in 2022, almost 30 years after the CBD came into effect, there is now genetic indicators included in the Global Biodiversity Framework on the Convention of Biological Diversity. And this is really important. So we have currently 196 nations that are parties to the CBD. So 195 countries and the EU, effectively meaning that all of these need to report on genetic diversity of wild species. So this has a huge effect on the countries and their biodiversity monitoring. So the text of the Kunming-Montreal Global Biodiversity Framework of the CBD says the long-term goal that genetic diversity within populations of wild and domesticated species is maintained, safeguarding their adaptive population. And again, the text for the target 4, which is the more recent or not so long into the future target, 2030, is that we have to maintain and restore the genetic diversity within and between populations of native, wild, and domesticated species. So this is quite strong wording, putting a focus on the genetic diversity of wild species. Unfortunately, not all 3 indicators made it in to the Global Biodiversity Framework, but 2 of them did. So the NE 500, so indicator 1, and the populations maintained, which is indicator 2. One of the strong assets of these 2 indicators are that they can be assessed without genetic data. So both DNA-based, but also non-DNA-based data. And I will come back to that in a second. But after the work to get the genetic diversity into the Kunming-Montreal Global Biodiversity Framework, no one was able to really rest, because then a lot of new efforts were needed in terms of trying to test and make awareness of the genetic diversity indicators, to ensure that all the guidance materials and documentations were there, enabling the countries to actually use the indicators. And also raising awareness, because all of the parties to the CBD, they need to report every 3 years on the Global Biodiversity Framework goals, targets, and indicators, and also produce their national biodiversity strategy and action plans. So efforts also went into raising awareness how to actually include genetic diversity in those plans. So what I'm going to spend the rest of my time here today talking about is one of these initiatives that tries to bring the genetic diversity indicators to use in countries. So I'm going to talk to you about the Genemo, which is an EU Biodiversity Plus project, which started a year ago. It's genetic indicators for nature monitoring. And it's quite a large project. We're a large consortium. We have 11 partner institutions across 8 countries. And what Genemo is set out to do is to co-create user-friendly workflows with country stakeholders in 5 selected countries, enabling them to report on the CBD Kunming-Montreal Global Biodiversity Framework genetic diversity indicators and use them for biodiversity monitoring and action. So we are a very diverse team. So we both have geneticists and natural scientists on board, but we also have social sciences. Because besides what I'm going to talk about in a second, how to test the genetic indicators, we also want to understand and co-create some outputs together with these country stakeholders in terms of enabling them to actually use the genetic diversity for both reporting but also use. So first and foremost, we have kind of 3 overarching goals. We want to determine the best practices to obtain accurate and robust effective population size estimates for species with DNA-based data, where we're building from or harnessing a lot of the already generated data that is out there in different databases, and again also building from the fantastic efforts of reference genomes and omics. And we will develop best practices to estimate NE from proxy data, so non-DNA based data, and the proportions of populations maintained indicated with publicly available data sources. And lastly, we want to develop standardized workflows to facilitate the calculation of genetic indicators in accordance with, of course, FAIR principles and open science practices. So DNA-based, you of course all know how to do that, but proxy data, so non-DNA-based data, could, for instance, be census size. It could be species population data, satellite data, so Earth observation data, citizen size data, etc. And it's easy to use with sufficient information, which is the key. The potential sources for proxy data, it could be species management reports, recovery plans, site inventories. It could be IUCN Red List, national species databases, scientific papers, consulting local knowledge holders, local NGOs, again, citizen science data, habitat quality data, field guides, and so on. There is a plethora of sources, but not all of them equally well in terms of the data that they hold. Some of them are quite patchy. So what I mentioned before is that we are working in Genemo together with 5 selected countries. So it's Norway, Sweden, Belgium, France, and Italy. And what we have done is that we have hosted in-country workshops to get an overview of the current situation in each country that is relevant for genetic diversity and indicators, and also what challenges the stakeholders are facing, and also the country as a whole, in order for both Genemo and the country stakeholders to follow a co-creation process to develop user-friendly workflows that can enable the countries to report on the genetic indicators to the CBD and also use them for biodiversity monitoring and action. And this is because we believe that by following a co-creation process with the country stakeholders, we can work on the outcomes that actually fit their needs and resources instead of believing what we think might be their challenges and also what they have of resources. So we actually get from their learning from their side what they need and what resources they do have to develop something that actually fit that. And in the end, we believe that by working in this co-creation, following a co-creation process, that will actually increase the chances of genetic diversity to be adopted and used by the countries. Some of the challenges that are common across all the countries are related to, for instance, species selection. So how to make the list of species that they should monitor in terms of the CBD, but also defining populations for genetic diversity protection as well. There are some QR codes which you can scan here, which is a recent effort from the IUCN Conservation Genetic Specialist Group, both the species selection guide, but also more recently a few papers on how to delineate populations using DNA-based data. I also want to highlight GENOA, which is a recently launched cost action network on genetic nature observation and action. Again, a QR code which you can scan, which will take you to the action, which runs in parallel with GENOMO, and the two complement each other very nicely. Also, a recent initiative I want to highlight was a team from the Conservation Genetic Specialist Group of IUCN joining forces to write a motion for the IUCN on a task force on genetic diversity to have that as a kind of a complementary approach to the red list. So that has also been put in and accepted and will hopefully become a resolution that the IUCN has to work on. And going forward, there will be joint forces between GENOA and GENOMO in terms of the countries, their national biodiversity strategy and action plans, and national reports to make sure that we get a lot of reporting and use of genetic indicators for nature monitoring. So I just want to end my talk today with saying that this is, we're building on a lot of efforts. There's been so many efforts and so many partners involved throughout the years to actually get to where we are now. We have indicators for genetic diversity. They're not perfect, but they are good. They are sound, and we are testing them, and they are usable, so you can use them globally, regardless of resources. I also want to stress that you can engage with an impact policy, but you have to be proactive and you have to be ready to put in a lot of time and efforts. And again, the more people you are, the more voices you have. So join forces if you want to go that route. But also, as mentioned, there's a lot of initiatives that work with countries on reporting and using of genetic indicators, initiatives that focus on the scientific work to actually improve those indicators, and also tools that can help ease the application of the indicators by the ones that are to use the indicators. And lastly, the countries, they are diverse. These were just what we're focusing on in GENOMO is 5 European countries. And even though you may think they are quite similar, they were not. And if we look globally, there will definitely be a lot more diversity. So they come with different starting points, different resources, different needs. But what is important here is to listen, to follow an inclusive process, to really learn what the countries need to actually help tailor some outcomes that can be relevant and useful for them. So this was my presentation for today. And if you have any questions, I am happy to take them now, or else I've put my email up in the upper right corner, and you're more than welcome to write me if you have any questions afterwards.